So our bodies are full of cells that are essentially stacks of chemicals undergoing chemical reactions. And some of those chemical reactions are taking large molecules and breaking them down into smaller stuff. And that's called catabolism. It's kind of like what happens in your digestive system, but it happens in your cells all the time. That often releases energy. You also have small bits and pieces of things, monomers, that get put together into larger structures like polymers, and that's called anabolism. And that usually requires energy. Anabolism, like anabolic steroids, right? Those are steroids that help you build amino acids into larger proteins. And all of those things together are called metabolism. Right? It's all of the chemical reactions that are happening in your body. We are constantly breaking things down, proteins breaking down into amino acids. Then we use those amino acids to build new proteins, and there's all this turnover that's always happening. And you can keep doing that as long as you have energy, right? You need a constant input of energy. Where do we get this energy? Well, from food. But most of that energy is wasted, right? The second law of thermodynamics states that the universe is always becoming less ordered, essentially. And what that means functionally when you're talking about chemical reactions like this is when you convert one form of energy to another, like chemical energy into motion in your muscles, or one kind of chemical energy, like glucose, into another kind of chemical energy, like ATP. Along every step, you lose some of that energy as heat, which is a very disordered kind of energy. So we're never 100% efficient. In fact, when you eat some sugar, about 40% of that is captured when we make ATP, right? About 60% of that is lost as heat. And that may seem inefficient, but that's actually incredibly efficient, right? That's more efficient than your car is, for example. And this is what that looks like. We are constantly building up polymers from our nutrient pool, and then when they are old or used up, or we need new things, we're breaking them down and reusing those nutrients to build new things, and round and round it goes. But you can't keep doing that unless you have an input of energy, right? And so we use some of the breakdown of nutrients for energy. We have to continue consuming new nutrients, so usually glucose for fuel. When we've been consuming a lot of nutrients, a lot of energy, we are taking those small things that we've absorbed, like fatty acids and glucose and amino acids, and we are putting them together into larger molecules for either function or for storage, right? So we're turning amino acids and building new proteins with them, taking glucose, storing it as glycogen. We're taking fatty acids and turning them into fats or triglycerides, where we store them for energy later on. And when we haven't been taking in a lot of nutrients, we go the opposite way, right? We take these triglycerides and we turn them into fatty acids, and then we burn them as fuel. We take glycogen, we turn it into glucose, and we burn it as fuel. We may even take proteins, turn them into amino acids, and burn them as fuel if we have to. This is called mobilization of metabolic reserves. So when you haven't eaten in a while, you may break down glycogen that's been in your liver or in your muscles, and you may turn it into glucose and use it. You may break down triglycerides that have been stored in your adipocytes, and you may burn those fatty acids that you produce to make ATP. And it all ends up kind of looking like this, right? We have storage in our adipocytes and in our liver cells for glycogen, and in our muscles. And as we burn through those things, right, we have to bring new stuff in through digestion and absorption, which you guys are now experts on from chapter 22. We're going to talk a lot about glucose in this chapter, because glucose is the primary fuel in your body for making ATP. And when you've eaten, you can store a lot of it as glycogen, which is just a polysaccharide, meaning lots of glucose molecules together. Making glycogen is called glycogenesis. Genesis means the production of, right? You can also break down glycogen, and that's called glycogenolysis, right? So this is a catabolic pathway. Fatty acids can be stored as triglycerides, or we can break down those triglycerides into fatty acids and glycerol molecules. Amino acids can be stored as proteins, or we can break proteins down to become amino acids again. So again, this is the two-directional pathway for each of these monomers into polymers. And yes, you can also build nucleotides into nucleic acids and break them back down. And that's important for, information flow, but that's less important for energy, and so we're not going to talk about it very much in this chapter. Okay, as we talk about catabolic pathways, I'm going to skip ahead a little bit to talk about glycolysis, which is the breakdown of glucose. This is not the order your book goes in, but I think it's important to talk about this first, because generally that's what you're starting with in your cells. Right? Glucose is the primary fuel of the cells, and so you start with a 6-carbon molecule of glucose, right? 6 carbons, and glycolysis is the process of breaking that in half into 3 carbon molecules, 2 3-carbon molecules instead of 1 6-carbon molecule. Now along the way, you are going to gain a net of 2 ATPs, and you're also going to get a couple of NADH molecules. Now these will be important later, but essentially these are carriers for high energy electrons. When I say high energy electron, I mean an electron that's further away from the nucleus. It's been juiced up a little bit, and that carries energy with it, because if that falls back down in energy level, it releases energy. You can imagine, like, sometimes that, if you have, dark, vinyl seats in your car or leather seats in your car, and a sunbeam comes in, right? That can juice electrons in that seat, but then they fall right back down again. But when they fall back down, they release energy. They tend to release it as heat, and that's why when your car's been sitting outside on a hot day for a long time, it's very hot. Anyway, so those high energy electrons are very important. So we end up with, we start with 6-carbon glucose, crack it into 2 3-carbon molecules, which are called pyruvate, and we capture a couple of high energy electrons in each NADH molecule, and we make 2 ATPs, which is really what we're here for in the first place, because ATP is the energy currency of the cell. And this is what that looks like. We start with glucose, and this just happens in the cytoplasm of the cell, just kind of wherever. And, I talked about it very simply. It's really a long process that includes 10 different steps, each of which has an enzyme that turns one intermediate into the next intermediate, and then another enzyme will turn that next intermediate into the one after that. I do not need you to memorize all those things. I just want you to know essentially what goes in and out of ATP. Right? What comes in is glucose, and we have to add a couple of ATPs to get the reaction going. But at the end, we get 4 ATPs. So we spent 2, we got 4. We get a net of 2 ATPs. We get 2 NADH molecules, which are each carrying some high energy electrons. And we have 2 3-carbon pyruvate molecules. There's still a lot of energy left in those 3-carbon pyruvate molecules. And if you don't have oxygen around, you can't take advantage of that extra energy. But if you do have oxygen, well, then you have a lot of ATP left over. So per glucose molecule, you probably get, in the low 30s in terms of ATPs from that. Only 2 of them have come from glycolysis, so clearly there's a lot of energy left to be gotten. Right? So glycolysis makes 2 ATPs. The next couple of cycles we're going to talk about make, up to 34 ATPs total. All right, skipping backwards in your book, but ahead in the process, we've now taken our 6-carbon glucose molecule, and through glycolysis, we've cracked it in half. We've gotten a couple of ATPs and a couple of NADHs from that, but we've got these 2 pyruvates. What do we do with those? Well, they will move on to the mitochondria, which, as you know, is the powerhouse of the cell, and that's where you get the rest of your ATP. So this pyruvate molecule is going to move in, and eventually we're going to want to dump those carbons into the citric acid cycle. But that 3-carbon molecule can't actually enter the citric acid cycle. We have to turn it into a 2-carbon molecule, right? So how do we do that? Well, this is sometimes called the preparatory reaction for the citric acid cycle, or sometimes it's called oxidation of pyruvate or acetyl-CoA reaction, whatever you call it. We take a carbon and we break it off of that 3-carbon molecule, and we make a 2-carbon molecule that's called an acetyl group or acetate. And that acetyl group gets shuttled into the citric acid cycle by something called coenzyme A, right? So coenzyme A takes the acetyl group, binds to it, shuttles it in to citric acid cycle. Well, together, that coenzyme A and that acetyl group are called acetyl-CoA, right? So you'll see that word, that term, bandied around. So then we shuttle it into the citric acid cycle. We'll talk about that in a moment. So, summary, we have our 3 carbon pyruvate molecules that we got from glycolysis. We pop a carbon off, make an acetyl group, coenzyme A shuttles it in to the citric acid cycle. What happens to that carbon? Well, it becomes CO2, right? This is why cells making ATP produce a lot of CO2, right? So the carbons that we break off become CO2, and ultimately they will get into our bloodstream, into our lungs, and we will breathe them out. Okay, citric acid cycle, very, very important. It's a cycle, meaning we start and end with the same molecule. Not that it goes in a circle necessarily like a dance, right? We show it that way, but that's just to visually help you understand that we start and end with the same molecule. And that's a 4-carbon molecule. It's called oxaloacetate. You don't need to know that. But oxaloacetate binds to acetyl-CoA, the acetyl group from the acetyl-CoA, and they make a 6-carbon molecule. That's called citric acid. That's why it's called the citric acid cycle, right? And then we go through this whole series of reactions, kind of like in glycolysis, right? And we've got all these intermediates and all these enzymes that help these reactions go. You don't have to know them all. But along the way, we pop off those 2 carbons that we added. We get a new ATP for each crank through, right? And we get 2 cranks through for each glucose, because remember we had 2 pyruvates and therefore 2 acetyl groups per glucose molecule. So we get a couple of ATPs from this. We also get a bunch of NADH molecules. And if you recall, NADH carries high energy electrons. We also get a couple of FADH2 molecules, right? Those are just, you can think of them as in your head as just essentially like NADH, because they are also carriers of higher energy electrons. And so at the end, we've added 2 carbons, we go in our circle, we've popped those 2 carbons back off, and we end up with the same 4-carbon molecule that we started with, oxaloacetate, and it's ready to take another acetyl group from acetyl-CoA, bind to it, go through that same cycle again, and over and over and over again, as long as you keep feeding it acetyl groups. What happens to those carbons that we popped off? Well, they also become CO2, which will move into your bloodstream and into your lungs, and we will breathe out. And you guys are already experts on that from having looked at the respiratory system. So now we've got 2 ATPs from glycolysis. We've gotten 2 ATPs from the citric acid cycle, right? 1 for each crank. And that's a total of 4 ATPs from a glucose molecule. And I promised you over 30. So where do we get the rest? We've used up all of our carbons already. Well, what do we have left but our high energy electrons, all those NADHs that we talked about, and those FADH2s that we got from the citric acid cycle. Okay, so we are going to shuttle those over. We're still going to stay in the mitochondrion, but instead of being in the mitochondrial matrix, just the center here where the citric acid cycle takes place, we're going to go to this inner membrane of the mitochondrion, right? There are enzymes in here that do something called the electron transport system. This is a closer look at the citric acid cycle. Again, you do not need to know all of these intermediates, but I do want you to understand how complex it is. And each one of these molecules, citrate, isocitrate, ketoglutarate, etc., your succinate, fumarate, malate, these are all intermediates in this whole cycle that starts and ends with oxaloacetate. Right? Again, I just need you to know the general picture, what goes in and what comes out. Right? What comes in is our acetyl group and our oxaloacetate. We pop off those carbons. We get a couple more ATP for each glucose molecule, and we get a bunch of NADH, right? 6 total, and 2 FADH2s total for each glucose molecule. So we have lots of high energy electrons to carry on to the next process, the electron transport system, which I will talk about in the next video.